I'm going to be talking about something somewhat different. I'm going to be talking about looking at microvascular retinal disease, but targeting it with a very new mechanism of action, which Ocunexus has developed. Basically, we have two compounds in development. One is called Xiflam, which is an oral compound, small molecule for the treatment of retinal microvascular disease. And then we have Peptagon, which is a peptide which was designed as a hemichannel blocker in order to treat microvascular disease of the retina. Now, we're targeting the inflammasome, which is a novel pathway that has existed as long as we've existed and as long as our predecessors have existed. And it is one that is responsible for significant damage to the microvasculature. And I'm going to explain how we get to the inflammasome and inhibit it. In order to do that, I have to talk a little bit about gap junctions. Gap junctions occur in the cells of the majority of our body. And each gap junction is formed by two forming hemichannels. Hemichannels are made up of proteins called connexins, and the one that we're targeting with our drugs is connexin 43, which is the most ubiquitous of those proteins. When these hemichannels are forming, they are generally closed, and they form one cell to the other, and they're joining cells in order to create a gap junction, which is the communication between the two cells. During a pathological condition, and it could be chronic or acute, the hemichannel opens. And once that hemichannel opens, ATP is released into the extracellular milieu, and that will activate the inflammasome pathway of inflammation. The inflammasome, in turn, will activate multiple cytokines and create the damage that we see, particularly in retinal disease, which is the ones we're talking about. Now, in order to evaluate all of this, we've done a lot of work, particularly cellular assays. And what you can see here in the top left is taking our peptide, which is a hemichannel blocker, blocking the connexin 43 to see if it inhibits ATP release. And what you can see in the top left, we've got basal, which is the black bar. The green bar is the insulted cells creating the open hemichannel and ATP release. And then treated with the Peptagon, we see it brings it back to basal. Now, these were treated with TNF-alpha and interleukin-1b, but we've done the same experiments with interleukin-6, with ICAM-1, VEGF, MCP-1, and IL-8. The next question is, okay, we can knock down the ATP release, but does it prevent the formation of the inflammasome complex? And what you can see in these cellular assays, in the micrograph A, what you're seeing, the small spots are the activated inflammasome, and these are identified by the biomarker NLRP3. On the right-hand side, which is B, these are cells that have been treated with a hemichannel blocker. We've knocked down the ATP, and you can see that the inflammasome has now gone back to basal levels. Xiflam is our oral drug, and I'm going to talk a little bit about that because there were a couple of things we had to do when we identified it as a hemichannel blocker. Number one, we had to show that it was equivalent to Peptagon, which was our peptide that we had used to do the cellular assays. And what you can see here is when we looked at reducing ATP, we find equivalence between Peptagon and the Xiflam. We also wanted to make sure that dosing systemically would not create a problem with normal gap junctions, and so we did a significant amount of work to show that at the doses we're going to be treating with, we will not see any problem. We then went to models of retinal degeneration. The first one is the bright light model. Animals are exposed for 24 hours, and they were dosed 2 hours into the light period and at the end of the light period. And what we see here on the left-hand side is the untreated animal, after 3 months of no further treatment, has a completely destroyed neural retina. There's also thinning of the choroid. On the right-hand side, where we were treating with the hemichannel blocker, knocking down ATP and the inflammasome pathway, we can see a well-defined neural retina. And we've also correlated that with ERGs, which we see at the bottom of the slide. And both for the A and B wave, we have distinguished visual function in the untreated animals and maintained visual function in the treated animals. We also took Peptagon into a retinal ischemia perfusion model for diabetic retinopathy. We wanted to see what would happen when we perfused these eyes and got leakage from these vessels. And we were looking for our target, which is connexin 43. And what you can see in that graph is we have significant overexpression of connexin 43, leading to ATP release, open hemichannels, and inflammasome activation. We also see astrocytic processes crossing the vessel, indicating that we do have endothelial cell disruption. And what the results are, we had 86% reduction in dye leak in the animals with the perfusion at a peak 4-hour versus the controls. And at the same time, we also had retinal ganglion cell density, which was maintained, so we had no loss of ganglion cells, essentially, compared to the baseline. We then went to an Akimba mouse, which is a model of diabetic retinopathy, and we looked at the Akimba mouse, looked at the retina to see if we had overexpression. And if you look at the green, you can see the overexpression in the Akimba versus the wild type. And then we went to human retinas. So we looked at donor retinas of diabetic retinopathy, also to see whether or not we had our target, connexin 43, upregulated and overexpressed. And sure enough, and you can see it red dyed here in the vessels in these diabetic eyes. So we've shown it in the animal as well as in humans. And what is the strength and differentiation of our technology? Number one, we validated the target. It's taken 8 years of work to do this. We've demonstrated that the target is present in the diseases of interest, both in animals and humans. We validated the mechanism, and we know that it's activating the inflammasome. We've shown that over and over again. And we also are addressing the activation of multiple cytokines, which we believe is doing the damage in either AMD or diabetic retinopathy. And right now, we're looking for financing to support an oral clinical trial, a Phase IIB clinical trial of oral Xiflam for geographic atrophy and potentially GME. We do believe that if we see a clinical result, this will be a game changer in terms of how we treat microvascular disease in both AMD and diabetic retinopathy. Thank you.